Welcome everybody to Drug Discovery Diaries, Season 2, Episode 4. We've been cranking these out. We have a huge guest with us today. It's a total pleasure, an honor to have somebody who, you know, I essentially had a big role in his existence and everything like that, but it's great to have John Lee with us here today, and there's a story behind the name and everything like that. We'll get into all those details. But before we get to John, first I want to shout out to my middle daughter. I'm showing another one of my Father's Day gifts. My middle daughter, who not only got married in May, but graduated from Johns Hopkins with her master's in biotechnology. So very impressed with her, very proud of her. But that's not as important as what Hillary has to tell us. When does the boy go?
He went.
He went?
He went.
Oh my goodness.
On last Thursday.
Last Thursday?
We moved him in. Yeah.
How was that? So John, Hillary's oldest,
yes.
went to University of Pittsburgh.
Yeah. And first year.
Yeah.
First time for all of us.
Did you cry?
So funny thing.
These are my questions.
Yeah, I cried on the 2 days leading up to it when he, was hugging our dogs and my younger son goodbye, and we were all sitting in his room the night before talking and stuff like that. Dropped him, unloaded him, and then because so far away, we stayed overnight, and then we saw him again the next morning, briefly, just
Right.
to say goodbyes or whatever.
He didn't like shoo you out. Come on.
He kind of
enough.
was doing that a little bit. So, we were trying not to be dramatic, and he barely hugged me. He was like one of these,
Right.
And then we left, and there was not a tear, and I was like, okay, this is much better than I thought. We literally got outside of the campus, and I lost it. It welled up, and then it was just heaving for maybe 10 minutes or so.
Right.
And then I got it together, and it was okay again. But this past weekend was difficult because, we feel like there's a piece that's missing. So it's, the house is definitely different,
Okay.
Yeah.
That's great. Well,
Yeah.
congratulations.
He had his first day of class today. He sent a pic of him with his backpack on, and he was, I don't know, he met another chem engineering student and they're
boy.
friends now, so.
Okay, good.
Yeah.
Where's he getting his beer from?
I don't know, but we think there were definitely shenanigans last night. Because we got a call around 9 and said, Mom, I have to take my roommate to the emergency room.
Oh, dear
And I
God.
said, you've literally been there for 3 days. What do you mean? And apparently they were playing Ding Dong Ditch with a couple of people in the hallway, a couple other rooms in the hallway or whatever. And Seamus went to open the door really quickly and smacked his roommate right in the forehead. And
Oh,
his
dear
roommate
God.
had a giant cut going down his forehead.
Oh, that's a good one.
Took him over to the whatever.
Urgent care or something like that?
Yeah,
or
and
campus?
yeah, on campus, and all they did was a butterfly stitch.
Okay.
And his mom and his sister or nurses or something were like, yeah, we think he needs real stitches. So Seamus drove him up to Olean, New York, which is the closest thing. And they sat there for 2 hours only to find out he really did not need stitches.
Oh, right.
And they put some more butterflies on him and they came back. So he was home about midnight last night.
Mm.
But I said, what are you doing? Can you relax a little bit?
Just wait till next week.
I know. So there had to be something going on.
That's awesome. No, that's a good first week. Yeah, I think it's a good first week.
Yeah.
Well, nobody died,
Nobody died.
and there were some bumps and bruises and shenanigans.
Yes.
Those are good,
Yeah. Yeah.
That's great.
So, yeah.
Outstanding. So we're here with John Lee, who is an executive at, is that the appropriate term?
I don't know. I'm waiting to see where this goes.
At SK Pharmteco. And as many of you remember, we had another executive from the same organization on a couple podcasts ago, Brian Tomkowitz. If anybody follows me on LinkedIn, when I announced that, you would have noticed that was our first video podcast. and John made the probably appropriate comment on LinkedIn about how aggressive it was, aggressive or risky to use Tomkowitz as the first face of. So this big chooch decided, he'd be willing to come on and take a stab at it. But you've been at SK for
4.5.
4.5 years.
Yeah,
We met in 2011.
that's right.
2011.
Yeah.
You interviewed for a job with me, and we had a target identification validation group back then, and I wanted good scientists. And so John's focus is on, well, not only is he a spectacular scientist, but he grew into the way I think the group eventually grew into cell and gene therapy, and then took a passion for cell therapy, and John will get into that in more detail. But John is a Pennsylvania native, and you can tell these details, but you had a mutual friend who also worked at J&J at the time. It might have been Centocor, but Centocor, J&J. And that mutual friend said to you, make sure you don't tell him you're a Cowboys fan. Because he's Michael, I was a big Philly fan. And it never came up, curiously enough. Because usually I ask people that. I tell people, I'm like, I don't care where you're from, who you follow, because it's important. It literally could make or break a relationship. And it never came up for some reason. I don't know if you just overwhelmed the conversation or what, but it never came up. I never asked you. And then after you were hired, it came up, and then you told me, and I'm like, you son of a bitch. Totally schooled me on that one. Are you still a Cowboys fan?
I don't tell many people these days, but yeah, I still root for the Cowboys.
All right, that's all right. We'll forgive you because the Cowboys suck. So tell us a little about yourself, your upbringing, and your daughter is how old?
She's 20.
She's in school.
She's in school. She did really well in high school.
Right.
It was one of these advanced programs,
Yeah.
going to community college, and she graduated with an associate's degree with her high school diploma.
Outstanding.
And so they, believe it or not, University of South Carolina transferred all of her credits.
Spectacular.
And so she is starting her second year of college, which is also her senior year of college right now. So
Wow.
this week she started, or last Tuesday was her first, and she's a senior in the business school.
Outstanding.
Yeah.
So she got her brains from her mother, clearly.
Obviously. Yeah, obviously.
Because my oldest daughter used to babysit her.
Mm-hmm.
I mean, just like for a week or something
Mhm.
like that. John had to go and play scientist for some time. So, yes, that's great. Well, that's wonderful to hear. All right, so give us your story.
You want me to start when I was like 4 or
You
5?
you
or?
can start wherever
Okay.
you want, because I'll bring something out regardless of where you
I'm
start.
sure. So out of high school, went into the military.
Oh, that's right. That's a great.
I'm a vet, United States Army, 13 Foxtrot. Used that time to kind of get my bearings on what I wanted to do in life, and then went to undergrad.
And you went into the Army because, when you came out of high school, you were like, I don't know what to do.
No idea what I
Really?
wanted to do. Nobody had gone to college, so there wasn't a whole lot of guidance.
Because you grew up in East Bumble,
Yeah,
right?
yeah, just outside of State College.
That is definitely.
Yeah, just outside of State College, and so it was the right thing for me in terms of getting some money for school and just getting a little bit of gathering myself and figuring out what was right. And so I used that time, decided I wanted to go into, I think like you, medical profession,
Okay.
and in Brian Tomkowitz for that matter.
Yeah,
That's
you're
what
right.
he
That's right.
professed too. And so I got in the back end of undergrad and decided I wasn't really sure I wanted to do that either, much like Brian had said, maybe for different reasons for me, and ended up going to graduate school, East Carolina University, to do cancer research. Whatever I did, I wanted to have an impact. So I did that, and then spent a few years in graduate school, got a PhD in microbiology, immunology, really was a cancer degree, cell biology, and then came here to Philadelphia to do my postdoc at the Wistar Institute. Did that with Meinhard Herlin, who's a preeminent melanoma researcher, and then on the back end of that, interviewed at J&J. Was there for 10 years. We could talk a little bit more about that, but then from there I went on to GlaxoSmithKline.
Can I interrupt you real quick?
Yeah.
When you were with Meinhard, did you know Meralda Bikiri?
I know the name. It was right before I was there.
Okay, she worked with Meinhard for several years,
Yeah.
but then she worked on my team at J&J,
Oh, she did?
and then
Okay.
at Century as well.
Yeah, she was there. I think she left right before I got there.
Okay.
And so then I went to GSK for a hot minute, and their cell therapy efforts kind of.
But you glossed over the whole J&J time.
J&J. Well, I figured you were going to ask questions about J&J.
Well, I mean, so let's get to the meat of it. First of
Yeah,
all, first of all, I have these resumes that get thrown in front of me, and I see one. I see one from a guy who has a good CV. His name is John Lee.
Really?
Oh,
Yes.
well, this is good.
His name is John, and I'm like, hey, little Asian guy, you know what I mean? And in walks this, I call him in for an interview, and in walks this big cabone. What are you, 6'4"?
6'5". Yeah.
6'5" with the heels on. Never expected that. And interesting fun fact, this is true, right? Your father was a jockey.
Yeah, he was 4'10". Yeah,
All right.
4'10". And the next question is, how big was the mailman or the milkman, right? That's typically the question.
I was actually going to ask how tall your mother was.
No, that's the more polite question.
Yes.
That's not where I used to go.
She's like 5'5".
Wow.
But my grandfather, my father's father, was 4'9", but my uncle is 6 feet. So there's this kind of weird,
That's
That's
crazy.
weird.
strange.
That's strange.
Yeah.
Yeah. Anyway,
Jockey.
what's
Let's
that?
get back to the jockey.
The jockey, right? But you came on, and I think back then we were doing some exploratory stuff, which was fun. It was a lot of fun. I enjoyed it. You had to work your butt off doing stuff because we were working mainly for the cancer DOS, and so that was a challenge from the perspective of what they were demanding and everything like that, and the personalities involved.
I have no idea what we're talking
Yes, no, of course not. But then we got an opportunity in, I think, 2014 to, sort of pivot into cell and gene therapy, which I thought was exciting. And you were, in charge of a group, or, had an opportunity to lead a group to essentially explore that.
Yeah.
That was, I think, your first foray into those things, even though you might have thought a lot about the platform and how it could be used. We actually were getting an opportunity, which was, I think, exciting. And J&J never did anything like that.
No.
That was way out of their wheelhouse, which came to bite us in the ass later on. Well, it just bit me in the ass. It didn't bite you in the ass. But it was fun. I thought that was fun. You had a good little crew.
It was probably one of the favorite times of my career, just because it was so exciting. We were building something, and we had carte blanche to, it wasn't just who you hired, it was also what we were doing. I remember sitting in your office reviewing papers very academically,
Right.
like, what do you think about this, and what are the pros and cons of going down this? And we ended up in 3 different areas, as I remember it. One was obviously cell therapy, another was AAV,
Right.
and then the last was around LMPs and
Yeah.
what we were doing there. And it was just kind of a fun time to kind of play in all those different sandboxes, if you will, and do something that was extremely early. I mean,
Right.
now everybody has done cell therapy at some level or these advanced therapies, but back then it was still very, very much didn't know. I remember
Right.
trying to transduce T cells and all the problems we had to deal with then.
No,
That
it
was
was-
elementary.
But it was still early days back then. I
Yeah.
mean, like the 2014, 2015 cell therapy was in its infancy. And I think the founders, the leaders of the field were still, even then,
Yeah.
trying to figure out how to do things well. So that was exciting. And then I think things took a little bit of a turn again, typical J&J fashion, where there was a desire to not pursue that at all. Until I think it was Carl June and Novartis got some clinical data, and all of a sudden it was the rage, because the J&J, they were fully invested in bispecific antibodies and
Yes.
biologics, but you couldn't argue with the data that was coming out of that other group and what they were showing, and I think there was a serious change of heart, I guess is the words to be used. But that was great, and I think, again, the learning experience, and navigating the interests of upper management at the same time as the science, it's always the challenge in big pharma. But so after that, and I had left at some point, but you had a chance to lead a group there for a while to get things going. What was that like?
That was great. It was, we had brought in Cilta-cel, Carvitke.
Yeah.
I guess that was like 2017. We did diligence on that, brought it in, and then, at that point, because we were building out an infrastructure, a framework to promote the clinical development of this asset that they were at that point fully vested in, and obviously we know the other side, how well that went. Then there was real discussion internally to actually do it the way that maybe you and I had envisioned very early on, that hey, now we've got this machine on the back end, this downstream manufacturing, clinical developments, all the analytical tools that we're building out. Let's build a pipeline.
Right.
And so it was a good time to be there, because you had all the support from leadership, and they were hiring, and we were outsourcing some work, and we did a collaboration with Fate Therapeutics,
Right.
and a few others that ultimately ended up moving through, and so it was a really great time. Really exciting, and we had, I think when I left J&J just after COVID, we had close to a dozen different pipelines in the program, and
Awesome.
it was a great time to be there.
That's the fulfilling aspect of it, I think, where you get support, and then also, like you said, you built a pipeline with the support of, the TA in particular. So that's great. And then, of course, the rest of bio, what was it called back then? J-Bio was gone, or?
No, that was still Janssen
Still-
Biotherapeutics.
So J-Bio, and so you had that machine as well to identify binders and go from there. And all the people associated with that, they were good people too. That's great. That's really good. So why the hell did you leave?
GSK was offering me a vice president role, and it was on the therapeutic side. So you understand that.
Yeah.
You guys have been there. There's always that little bit of abrasion between what was happening on the discovery side in J-Bio and what was happening on maybe the discovery development side.
Right.
With the TA, and I think we navigated that pretty well, to be quite honest. But it was just a good transition for me. It was a title,
Right.
A VP of Big Pharma was a title that obviously you always are striving towards, and that opportunity was there, and it was an opportunity to do some different things with another big pharma company, and so I felt like it was the right decision for me to kind of spread my wings and get a little more independence and experience.
So you went for the money.
I went for money.
Don't we all? Come on.
That's great. Did anybody go with you? I don't remember if anybody, did you ever take anybody with you?
I had a number of a number of conversations, but we never made it official. I had a lot of people join me at the Center for Breakthrough Medicine. We were going, I don't know, but I had a number of people join us there, but not at GSK.
Okay. Yeah, we'll talk about the Center for Breakthrough Medicine. So how long were you at GSK then?
Only a year. Yeah. So when I got
I thought
there,
it was longer than that.
it was a quick period of time. So when I got there, I reported into the global head of oncology. He left maybe 90 days after I got there, and so then I was interim reporting to the chief scientific officer,
Right.
who maybe 60 days after that he left
Dear God.
for a Jeff Bezos company.
Okay.
And so at that point there were 2 layers, and there were a lot of questions, and obviously we know what happened with GSK after I left in terms of their cell therapy. They basically triaged it all, and so at that point in time it was, for me it was like, okay, I can stay here and see this out, or I can just maintain momentum and do something else.
So backtrack a second, when you were at J&J, you had your hands in obviously the discovery aspect.
Mm-hmm.
Did you, I'm assuming you also had your hands in some of the manufacturing components.
Yeah, we worked pretty closely with some of the API folks.
Yeah.
We did, and this is something that's very, very relevant. We sat down and said, okay, on the R&D side, what can we do that is completely manufacturable on the other side? So when we throw something over the fence to the CMC team, they don't have to develop it, it's already developed, right? And so we sat down with them at some stage during the genesis of my team and said, okay, well, here are all the different unit operations within cell therapy, autologous cell therapy manufacturing. These are the things that make sense for us just to do on the R&D side, so that we don't have to worry about developability on the other side. And so we worked very closely with the team. You would know some of the names to put that into place. And at some level it was a little more expensive. We were using G-Rexes, for example,
Sure.
in the R&D lab, which is kind of overkill. But if you think about what it costs, it's a pittance compared to what it would have been to transfer out of flatware into the G-Rex and develop all the parameters there. So we just decided early on to really focus in on developability on the R&D side, such that when it did go across, there was a pretty streamlined route to getting into GMP.
Now that makes, that's something that's a growing pain, or something that on the antibody side, J-Bio or its predecessors went through a long time ago, because the concept of throwing a molecule, like literally just, here's a sequence, go and take this from here, and the time spent and the knowledge that had to be gained in order to make it developable was expensive and time consuming, and it so just made complete sense to do some of those things when you picked the molecule. In other words, look, it's all ready to go. It's translatable to your platform, and it sounds like you guys did the exact same thing, which makes makes things go smoother. That's for sure, quicker and smoother, which is always the drama with in big pharma. How do you make things go quicker and cheaper to some extent? So that's spectacular. Okay, so then you had an opportunity to go at GSK. You took it, there for a year. That was COVID, right?
That was during COVID.
So I remember talking with you, and you literally had responsibilities in the U.S. and the U.K. Is that correct?
My entire team was, I had one person in Collegeville, outside of Philadelphia, and then 40 people in Stevenage, UK.
Dear God.
So I would get up early, they're 5 hours ahead.
You're always front loaded,
and then all my clinical, because I had the NYESO assets that went back to Adaptimmune, but was Letacell. All the clinical development colleagues were on the West Coast, so I had like an 8-hour time difference. That was it was.
Did you sleep at all?
Not much, not much.
And did you, because it was COVID, did you do much traveling?
No, I mean I wanted to. I mean,
Right.
when you're building a team, you want to be there with them and really getting to know people better. But at the time, England had a 30-day incubation period or whatever they called it. And if you flew there, you had to wait in a hotel for, I think it was like quarantine, that's the name of the, so you were waiting for a long period of time. It just didn't make sense. So,
That's horrible.
Yeah.
That's horrible. That would be enough
They
to
were
piss me
stricter
off
than we
too.
were.
Yeah.
That'd be enough to tell me to look elsewhere, you know?
I mean,
Not to mention, you know, the fact that they dumped her.
Yeah.
But the NYSO was a TCR.
it was a
Okay.
TCRT program that they had ultimately wanted to take into lung cancer. The reason they brought it in, they in-licensed it from Adaptimmune. They brought it in to see whether or not it would work for non-small cell lung carcinoma.
Right.
And so they ran, you know, obviously a trial for that, but then it was working for synovial sarcoma, but
Right.
synovial sarcoma is maybe, what, a couple dozen patients?
Yes.
Yeah, small indication.
You know, and so it didn't make a whole lot of sense to do it for that. And without sharing too much, there wasn't a whole lot of reason to move forward around lung.
All right, so then you explored your opportunities,
I did.
and somehow you landed at Center for Breakthrough Medicines, which is, you know, again, it's a local organization,
Yeah.
more manufacturing focused, and you got a gig there.
Yeah, so what we skipped over a little bit was when COVID happened, I decided between J&J, not between, but started with J&J and ended at GSK, I enrolled in an executive MBA program
Oh, aren't you sweet?
to understand a little bit better. You know, I seen a lot of drugs, especially when we were at J&J, like good drugs that went to NME and then just never went anywhere.
Right.
It was frustrating. Like,
Of course.
why? And you know, obviously there are a lot of smart people there, and I thought, well, there's got to be more to this than what I'm seeing. And so we were all trapped inside for a year, so I figured, why not do this? So I enrolled in this program to want to learn more about the business side of what we do. And I finished that up at GSK, and then this opportunity was afforded to me through CBM to really kind of get into the black box of cell therapy, and that was around manufacturing.
Right.
It's a piece that, you know, even despite what I've already told you in terms of what we did in the R&D side, was really still relatively unknown at that time. I mean, I think you can still make the argument that we're still learning, but
Right.
so it was an opportunity for me to learn a lot more about the GMP side of cell therapy, to kind of fill that gap, and then also flex some muscles around, you know, running a business and,
Right.
and having that with, you know, the ultimate end goal of, you know, one point maybe being CEO or CSO of a cell therapy biotech, you know, and having that kind of end-to-end experience.
They didn't make you stay after paying for that, I'm assuming?
Oh, they made me pay it back.
Oh, you're kidding me.
No.
Oh, God.
They made me pay it back, but I wasn't sticking around, you know, just to watch them shut it down. It didn't make
Okay.
any sense.
All right, fair enough. Fair enough. And so the experiences through that program were valuable.
For sure.
Okay.
Yeah. I mean, it's like anything, right? I mean, even the bad things that you learn in business and in your work environment still teach you valuable lessons about what's next.
Right.
So most certainly, you know, I've learned from everything that I've done.
Okay. Good answer. Good answer. So then CBM comes along. Had they had any sort of start in the cell therapy manufacturing arena, or that you kicked it off?
I kicked it off, so I was
Wow.
employee number one, and
Employee number one.
employee number one on the cell therapy side, sorry.
Oh, okay,
Oh,
okay.
Yeah, sorry, that's fair. I was about employee number 200.
Okay.
But they, you know, they started off in the analytical side, and then viral vector came online, and then cell therapy was next. And so then I built out a team, very much like what we did at J&J, you know, building out the team, SOPs, training, etc., all the things that you do when you're building out GMP. I was even designing and building out the GMP facilities
Wow.
that were there, and did that for about 18 months. Along the way, we manufactured some material for several different customers, and then SK Pharmteco at that point in time came in. They were the Series A investor, and they executed a strategic option to become the majority shareholder. And at that point in time, I was asked to leave the cell therapy role and go into more of an executive role, as you called it.
Oh, that's about
in
right.
the lead-in, which is running the business unit. So now I'm on the commercial team. Obviously still wear a technical hat when I need to, but do a lot with strategy and thinking about where the market is heading, in terms of advanced therapy, and making sure that the things we're doing as a business are aligning with not just today's needs, but the needs of tomorrow.
Fascinating story. To think, he went from a lab at Wistar to, right, as a Wistar, right?
Yeah.
Chopping heads off mice and that kind of stuff.
Gassing.
I'm sorry.
I'm sitting here
I
like.
know. Well, because, I have a story, but I'll save that for private
That was a
conversation.
long time ago. You're dating yourself.
I got a story, but I'll leave it alone. To an executive, thinking about, and like in, let's see, this is 2026, so 15 years,
Yeah.
right?
Yeah. Life happens fast.
You think you're pretty great, don't you? You're pretty full of yourself, aren't you?
This is what I was waiting on when I showed up.
No, it's great. It's a great story. I think that, even with Tomkowitz, the conversation about learning, it's so important to learn multiple sides of the business, so you got a strong foundation on the discovery side. Everybody takes their lumps there too, especially when you reference these things where you start programs and they're going well seemingly, and all of a sudden they're cut, and it's a big kick in the ass to have to deal with those things. And it's like, why? And they give you some reasons, and they all kind of make sense or whatever. But I think appreciating that from the perspective of not the science side, but literally it's like you're in a business, which is heartbreaking to some extent when you think about the consequences of that, especially if you were involved in a program that was doing well and had the potential to do well. This is a little bit of a pissing match on the business side of things, but they gotta keep things going, and ultimately it comes down to it. And then you pivoted, and you got that experience not just through the MBA, but on the manufacturing side of things, which is something I used to complain about when I was J&J all the time, because there was. We talk about throwing things over a fence. There was a lot about that. There was a lot of that going on, and you could not get that exposure.
That's right.
And you need to have that exposure to complete the picture. Why it's important to do the things, some of the things you need to do, and you learned them as well, to make a product. This is something that comes up all the time in the startup world. It's like, great, you cured a lot of mice. In the lab, is this a drug or not? Or is this a product or not? And it comes down to some of the things I think you learned, which were, can you manufacture this?
I think that's also the great thing about startups, is because it's such a condensed group of people that you learn very quickly all sides.
Right.
Development, regulatory, manufacturing, because you're all
Sure.
talking to each other constantly.
Right.
So it's very, whereas big pharma, it was totally hurling it over a fence. You never saw it again. And then next thing you know, the clinicians that do a great job, but they're getting the awards for developing this drug and everything. And you're just like, but I worked on this for 10 years.
Right.
Like, I don't need an award. But, at the same time, it's like, aren't we just one big team?
Right.
But you don't get that feeling the way you do when you're at a startup.
True.
Yeah.
Names that go in the papers, after the clinical trials come through, where it's not the discovery team.
Mm-hmm.
Maybe the discovery CD compound or the discovery team leader might get involved in those things, but oftentimes it's not. Not that I care or you care, I'm sure, about those things, but it's not important. But it's a big, big piece of the picture that, you think about in the grand scheme of things, discoveries like this and the other stuff is like that. And it's a cross to bear. I used to tell people all the time, because people used to complain all the time, why are we not getting recognition and all this kind of stuff. It's like, look, you're in discovery.
Yeah.
You have to learn to live with this and accept it, or get out of the business.
Exactly. If you want recognition, go to a different part of
That's part of the
it.
organization. You know, you just have to pat yourself on the back and move on to the next program, you know, and, but that's the way the world is. You mentioned, you know, I think the future, because cell therapy, we lived it from a different perspective, has gone through this type of world where everybody was like, it's the future, it's gonna solve everything, and or maybe not everything, but you know, that's where people were investing a ton of money, certainly in the late 2010s, and then up until, you know, 2021 maybe, you know, and then it crashed.
Yeah.
And your thoughts on why maybe that happened?
I mean, I think the primary reason is it wasn't generating the returns for the investors that they had envisioned when they were dumping all that money. When you talk about curative therapies for diseases like cancer, of course that's exciting, and so you got a lot of people, a lot of investors taking money and investing it into these curative therapies, and they are indeed curative, but the financial models we've learned, it's really tough to make money given the complexities of the cold chain, the manufacturing process, just the logistical hurdles, and all of that, especially for autologous, gene-modified autologous cell therapies, has been difficult to kind of navigate. And so, investors go where they're going to get the return on their investment as quickly as possible. I still firmly believe that cell therapy is the future of medicine. I really, in my heart, believe that, because cells can do things that small molecules and biologics can't. We've got billions of years of life on Earth to kind of substantiate that hypothesis. But right now, and I think I heard you talk about this with Tomkowitz when he was here, it's like, it's cyclical, and what we're going through right now is certainly a down period for cell therapy. That doesn't mean that there isn't good work happening. There absolutely is. I mean, we could talk a little bit about maybe in vivo CAR Ts and what's happening there, and some of the the excitement. But there's also good work happening in the regenerative space. There's good work happening. TILs have had some pretty good success in melanoma, in the solid tumor environment. So there's still good stuff happening. But right now, yeah, it's tough, and I think it all goes down to it's not the science. The science will win, but it's money right now.
You think about the conversions of cell therapy with already established therapies, such as small molecules and immune checkpoint inhibitors. I mean, these things might already be on the market, but could make certain cancers more sensitive to cell therapy, especially solid tumor, obviously.
I think we saw that. I mean, wasn't it just? I mean, you guys might even know more about this than me, but just last week I saw there's a new RNA vaccine for melanoma.
Yeah.
Where they're combining with Pembro, and I mean, it substantiates exactly what you're describing, right? Where you're bringing these things together, to bring out the best biological attributes of each of those modalities to, obviously have the maximal benefit for the patient. So it absolutely is where all of this is trending, as being cell therapy centric, though I still wonder how much we've really extracted from what I think is a very powerful modality because of some of these more logistical and monetary hurdles.
I think that you brought up two points that I absolutely 100% agree with, which is rare between us.
It is.
So one is, I think cell therapies are a spectacular modality or platform or whatever because of everything you just said, which is it's the most complex entity to be used as a therapeutic. In other words, you're not talking about one or two or even three, depending upon the type of what you're comparing it to, pathways you can influence with a cell because of how complex it is. Now it's got the baggage of, you have to understand a cell in order to be able to take advantage of them, and not just the good but also the bad. but I've always been, and that's why, like, I think, you know, we're seeing a little bit of a turn towards regenerative. I think regenerative opportunities, even our former company, I think they went on, I think full, you know.
Autoimmune.
Oh, no,
Sorry.
type
Diabetes.
1 diabetes.
Yeah,
In
sorry.
other words, you know,
Mhm.
create a beta cell and
Yeah.
those kinds of things. And I think there's a future for cell therapies in that arena as well, a big one. You know, even like my opinion is like iPSCs in particular. I think, yeah, that's where they should be applied. You know, if you can make a cell from an iPSC that does something like this native cell in that arena, it's a great application. But the other thing you mentioned is, you know, the cyclical nature. I have this slide that I used to use over and over again because, I mean, cell therapies have been around for maybe 10, 12 years, 13 years, something like that, maybe 15 years.
As we know them now.
Like the CAR T type cell
Sure.
therapy, right? And so that's not a long time when you think about things. And you know, I think about antibodies. You know, I got my start in the early 90s, and I mean, you remember. I mean, how old were you in like 1988?
14.
14, okay,
Yeah.
so maybe you don't remember. But, you know, the antibodies were the cure for everything.
Mhm.
Monoclonal antibodies, you know, they were gonna magic bullets, all this stuff, hyped out the wazoo, and then they came crashing down
Mhm.
for a number of years. And then slowly, and then, you know, they had their ups and downs and that kind of stuff. And, you know, and it's the same thing that monoclonal antibodies had the same challenges. They had manufacturing challenges. You know, they had, you know, specificity challenges. They had rejection and PK challenges and that kind of stuff. Well, it took another 20 years to figure all that stuff out. They figured it out, and so it's just, you know, where they are now, and they had a resurgence as a result of some of the concerns associated with cell therapies. That you know, they found another gasp of air to get going again. But it's the same stuff. It's like any other platform. I mean, even small molecules to some extent, right? Oh yeah, you got a great enzyme inhibitor. Well, can you manufacture it? Is it? You
It's
know,
still evolving too.
Yeah.
You know, large molecules, and you know,
Yeah.
now we have multi-specifics. And it's like,
Right.
okay, what kind of arms can we put on here, and
Right.
let's hook it up with the cytotoxic and make an ADC, and
Right.
you know, it's constantly.
But even that, like that, I mean, that's a great point, right? So like ADCs, I remember when I started at J&J, we were exclusively biologics. You know, I was talking with Seattle Genetics was one of the bigger ones, and they're like, oh, ADC. Nobody at J&J wanted to do ADCs at the time. It was
Right.
all, you know, either T-cell redirection or just,
Yeah.
you know, standard, you know, antibody. And at some point, ADCs came back.
Right.
And, you know, so everything happens very cyclically, you know, and that's why I said at the beginning that science always wins. We figure things out.
Right.
It may not be on our own timelines, but science will pull through, and
Right.
the biology will push these different modalities forward.
So what do you think the next best thing or the next new thing is needed for cell therapies?
Oh wow! I mean, that's I mean, I think we need to find out what's happening with the in vivo CAR Ts. I mean, if that works, if it does work, I mean, the clinical data are there to suggest that it does work. I have some reservations around, you know, injecting systemically lentivirus and hoping it goes to the right place and that there are no adverse events.
Indeed, yeah.
I mean, we'll see. The jury is out on that. But if it works, then I think that is the next kind of step. And there are a number of players in that space. But I think ultimately what we're going to see is kind of a balance of different approaches. And what's going to enable autologous cell therapies is a number of different factors. You know, we're going to have to get the healthcare systems to kind of support paying, and commensurate with that is bringing the payment systems down to a level that that they're willing to pay.
Yeah.
Right.
And how do we do that? Well, maybe alternative cell types. I know you guys obviously have some experience there with iPSCs and what that might have, the potential it has. Automation is a big area. We just saw some big news this week with one of the bigger players in that space, Solaris, and one of their big people pulled out, and I guess time will tell as to why, whether it was inherent to the platform or if it was more inherent to the company that did pull out. But there are a lot of automation platforms that I think are really good. Cellular Origins has some good stuff. Multiply Labs is working on some things. Ori Biotech is another. All of these players, the number one cost driver in a manufacturing suite is people.
Right.
Pull the people out, all of a sudden it gets a lot cheaper. And you know what else people do is they make mistakes.
Right.
So if you can pull them out, deviations go down, so the cost goes down. And there are a lot of industries where automation has taken over. We don't have to name them, but I mean, the automotive industry probably the most prominent one. And as we start to standardize the approach, I think you're going to see more of that, and that will bring down the price, and we're going to end up seeing kind of a return to some of these more traditional cell therapies.
Okay.
Yeah.
What about like in oncology then? I mean, there's still some drama associated with a cell therapy, whether it's ex vivo, autologous, iPSC derived, or even in vivo with your lentivirus or your nanoparticles.
Mm-hmm.
There's still some technological challenges associated with tumor microenvironment, those kinds of things. What are your thoughts on that?
Yeah, I mean, this is probably an area where the science still needs to be drudged up a little bit. I think there are a lot of different ideas in terms of think about trucks. This is probably one of the early stage, but it's a good example where cells possess the capability to do more than just kill. They
Right.
can accentuate the tumor microenvironment or modulate the tumor microenvironment in such a way that it makes it more susceptible to whatever it is that you're delivering, including some of the the ancillary, maybe small molecules or biologics. And you could even get a cell to deliver a biologic,
Yeah.
right? So there's a lot of different things that can happen there, but I think it's probably still early stage with
Okay.
regards to what's going to win out. I think it's like anything else. What we've learned is somebody finds something kind of cool, and then everybody iterates around that to get to a place where it validates or substantiates the path forward.
Okay. Yeah, it's complicated. I mean, I think it's, especially for solid tumors where you have that situation and not well or even completely or even close to completely understood. And then how to, I guess, go about addressing those things. Lots of cool stuff that comes out of academic labs. I think that maybe give you a little bit of hope, but somebody's got to sort of take the reins and make the case for it to be funded and then tested really where it counts, which is in the clinic.
That's the hard part too, right? I mean, so many great ideas, but no funding.
Right.
And it's not everywhere that's like that either. Philadelphia's especially bad right now, I feel like. But you look at places like Boston and San Diego, they're
Yeah.
thriving, the biotech space. So,
Yeah.
that comes first. We
Yeah.
can't really put those good ideas into action unless, there's the right funding there.
Yeah, I mean, I think what I see a lot of, I was at JP Morgan this year, on the business side, but, underlying that obviously is the science. And 80% of the people I spoke with, young biotech, early stage clinical, were CEOs asking, what our presence is in China.
Oh, yeah.
And these investigator-led trials, which right now are kind of hot because of this past week we learned there was a death with an in vivo CAR. It wasn't necessarily it wasn't lenti. It was a LMP circular RNA, as I understand it. But these Chinese investigator-led trials, I think, they're super hot. I think I read recently there's 14,000 different
Jeez.
trials that are underway. And it's really drawing a lot of these, let's call them modestly funded biotechs to China so that they can develop their drugs there, get clinical POC, or, it's fast kill, or,
Right.
Fast survive. You get to that data inflection point, or the value inflection point that's mediated by the data, and now you can come back to the States and say, to investors, look, this works, and now we can move forward more quickly. And, frankly, it's not a political podcast, but I think our politicians should be looking at this and saying, it might make sense to implement something, maybe with more severe or higher guardrails in the US, because as that infrastructure begins to leave, it becomes more difficult to bring it back. And I think we can all agree that that's not what we want.
I couldn't agree more. I think that's the one of the biggest challenges is investors, like you said, John, they're looking for the quick kill or the quick win. You can't get that here as of now. In other words, the hoops you have to go through in order to get into the clinic, even if it's with an IIT, a U.S.-based IIT, it's tremendous, and for the right reasons. In other words, right? Patient safety.
Yeah.
Maybe in certain indications, it's not as, this sounds harsh, but not as big a concern because these people, some of them are going to die anyway unless they try something. But nonetheless, they'll be left with that drama to have to deal with. But you can't compete. They're just pushing things through into humans without the regard for the patient necessarily at heart. And that's a debatable commentary. Like, how do you compete with that? And so investors are like, trending towards those organizations versus whatever you can do here. Yeah, it's a real challenge. It really is a big challenge for startups. And I think the U.S., like you said, I think the government has to put a lot, like you can't, what's the point of, there's no even point of innovating. If, you can't innovate without some financial backing. Maybe a company can say, a US-based company can say, we're going to do all the trials in the US, but, in China or something like that. But it's just, it's a huge hill to climb.
Even like preclinical development is moving to China. We're not just talking about
That's
clinical.
right.
We're talking about like preclinical manufacturing.
Right.
It might be an idea here,
Right.
but then it's bare bones company structure,
Right.
very lean, and then farm everything out.
Right.
And it's killing I know firsthand, it's killing the job market.
Right.
But it's becoming more and more like that. So, yeah, we either can be a front runner in the world for science, research,
Right.
developing products, or we just won't be.
Right.
And people have to be okay with that, but it's,
It's tough to imagine.
yeah.
It's tough to imagine
It
that
is.
that's what it's come to.
That's why we just want funding for scientific research.
What's that?
We just
Yes.
want funding for
That's
scientific
That's
research.
right. Nice. Very well done. I'm proud of you. Okay. I think that's, so what's next for you?
I just plugging away,
Okay.
continue to do what I do. What's next? Trying to get SK to a place where, we file IPO and
Right.
you live that monetary dream that you referenced earlier. And, from there, we'll see. We'll see where things go.
Is there talking about cell therapy, we could talk about that the entire time, but is there
Yeah.
something else that, intrigues you, or a field that you're watching in particular?
I'm still very near and dear to cell therapy. I think there's just so much untapped potential. So I still, if I had my druthers and, whatever happens for me next, I'm pretty sure it's going to be in the world of cell therapy. In what capacity? I mean, I think it could be any number of things. I think it could be in consulting. It could be in, C-level and running a small biotech. I think, there are a lot of different areas. But, as long as it's cell therapy, I think I'd be more than happy.
It's a very politically astute
Yeah.
answer to that question.
Yeah.
I don't know if, who else is watching.
You never who's that. We'll have a beer later, and I'll tell you everything.
That's great. Well, this has been wonderful. I think that, I don't know how you feel, Hillary. I think I know how you feel, but I mean, I've always been a cell therapy fan as well. I mean, again, for all the things we talked about, I think there's a tremendous untapped potential there still. And so I would like to see it succeed. Some of the things we talked about with regards to how it can be applied, and some of innovations and discoveries that I think would really, really give it a leg up, again, against this wall of having to compete with countries and the need to generate clinical data as quickly as possible, make it a challenge. But I would love to see it go places too, because I think it's, like we talked about at the very beginning, there's nothing more complicated yet fascinating and with such potential as a mammalian, especially a human cell, for what it can do. And I think there's still a tremendous future for it. Still early days, man. Still early days, It's just the beginning of, I think the wave. Would love to see it have an impact on solid tumors in particular, for sure. And of course, going into autoimmunity made sense, but that was sort of just a simple pivot.
Yes, I feel like it was a little bit of a bridge to keep cell therapy alive for a lot of companies, and whatever, it makes sense. It's helping patients.
Right.
That's the end goal, right?
Yeah.
But for sure, everyone started off with CD19 directed CAR Ts. It was the easiest, lowest hurdle, and it's understandable you start there, but you gotta expand.
Well, I mean, just in general, eliminating a cell type like a B cell, we can live with that. But it's when it gets a little more complicated with other target cells, it gets a little gamey. But I have faith, like you said, John. Science will take us there, and science will win, and that'll be the future. Closing remarks, young fella.
It's it's great to hang out with old friends. That's my.
You consider Hillary a friend?
He is
probably
now.
more than you.
I'll be nice.
Dallas Cowboys fan. That's a goddamn disgrace. How do you live with yourself?
And I call you a friend.
Unbelievable. All right, this has been great. Hillary, anything else?
No, but you're lucky you have me tonight, because I got a filling this morning.
Okay.
I never had cavities in my life until I turned 40, and now all of a sudden,
Why are you
I'm
telling us
getting
this?
because they were just supposed to, numb the one part.
Yeah.
Well, I guess some weird nerve was attached to a bunch of other ones, and my whole mouth went numb.
You're kidding me.
No, and I said, "How long is it going to be?" How long is it going to be? And she said, "Just a couple hours." And this was at 7:30 this morning, so I was like, "Okay, I'll probably be fine by this afternoon." But it literally, I'm trying to suck on a straw to get something to drink.
Though, she should have been like Kramer and.
That's right.
With Jerry's kids or whatever.
Yeah, that's exactly right. Exactly.
I was
what
like, "Can I
thinking.
drink coffee?" Can I drink coffee? Yeah, sure, you can drink coffee, but you
Suck
might
it to
not
a
feel the
straw.
temperature. Don't burn yourself. Stick your finger in it first. And as I'm trying to drink it, it's like, dribbling.
What a way to end.
Yeah. It's a visual.
It's a visual. I was gonna say, it's like you should have took pictures.
I was smiling on the inside, being all over the same.
Oh my God. Oh, that's unbelievable. All right, well, this has been great. John, thank you for coming and by and talking with us. It's been a pleasure, lots of insight. That's okay. And I think, like I said with Tom Kowitz, as things progress, we'll love to have you come back, and
Happy to come back.
we can transition from just an overview and catching up to you know, a pointed topic. In other words, hey, here's something that just came out, and we'll go
Yeah.
from there.
Let's do it.
Excellent. Thank you, everybody.
Yeah.
Thanks, John.